Hi everybody, so this is going to be another mini lesson on gene interactions, and specifically with this lesson we're going to focus on epistasis. The prefix epi means upon or over, and then stasis means standing. And basically what this means, if you recall from the one lesson that we did in class, a perfect example is the Labrador Retriever, and how there's a black lab, chocolate lab, and yellow lab. So those are like three different phenotypes for coat color, but it's actually controlled by a couple of genes where basically one gene will mask over the phenotypic expression of another gene. In this example here is pigeons, and so basically pigeons have these different patterns. They could have a T-check pattern, a check pattern, a bar pattern, or barless. But there is another allele or letter combination, gene combination, that would, if present, in this case, both of the genes or alleles have the spread bird gene, that would mask over all of it. So it's almost like if you were to like color a pigeon and give it some different types of shades and patterns, and then take a black crayon and color over all of that. That would be like this example, where the gene will actually hide pattern. Another example of this is with llamas. So I'm going to play just a little bit of this Amoeba Sister section of video that talks about how epistasis occurs in llamas. Again, this is when one gene depends on another gene for it to be expressed.
Epistasis is when one gene really depends on another gene for it to even be expressed. I mean, it really depends on this other gene. So let's pick an animal like, okay, a llama. Let's say this llama has a dominant big B allele, which means its wool will be black. So big B, or big B, little b means that it will have black wool.
So just a little review here. The big B, that's homozygous dominant for black. Big B, little b, since they're two different letters, or they're the same letter but different cases, that's what we call heterozygous. Just a little review there.
And let's say that if a llama has a pair of recessive alleles, little b, then it will have brown wool. Now, what if there is another gene that controls whether this pigment will even be expressed in the llama in the first place? In this hypothetical example, a llama could have this other gene coded for by big C, big C, or little c in its genotype. However, if a llama has the genotype little c, it will not allow the other gene for wool color to even be expressed.
So again, if this little c is there, as we can see in these three llamas, what that will do is it'll mask over the expression of the other phenotype.
Since we have two genes here, the gene for wool color and then this gene that controls whether the color is even expressed, this calls for our favorite 16-square dihybrid cross. If you notice in this dihybrid, crossing two heterozygote llamas, any llama with big B and big B, little b will typically give a black llama, and little b will typically give a brown llama, in all cases unless they inherit a little c. If the llama has a little c in its genotype, then the gene for wool color is not expressed, and the llama is albino. This means that no pigment is expressed at all. Pretty interesting. There are many other
Okay, so that pretty much sums up our gene interaction epistasis. Here is another example, explanation, where an allele of one gene, again, spreads or like it masks over the visible phenotype of another gene. So we have your typical gene of T-check, check, bar, and barless, and then this epistatic gene, the red gene, is epistatic to all the pattern alleles. So when they say it's epistatic, it means that it's going to mask over over it. Okay? All right, so that's it, and we'll move on to the other one.